Okay, we're in the home stretch. We have 4 presentations to do. And we're going to start with bringing back our European scholar, Hannah Douglas. Hannah, are you here? Okay, come on up.
So hello everybody. I really cannot see anybody at all. My name is Hannah Douglas. I am the 2022 European Our World Underwater Scholar. Last year in August of 2022, I graduated from my undergraduate degree in marine science. In my final year, I specialized in benthic ecology, which is essentially the study of things that live in, on, or around the seafloor. And although I specialized into anemones, I have a real fascination with all walks or crawls of life in the sea, plant and animal. My objectives for this year were to continue studying life in the seafloor while building up my own skill set to be able to do so more safely and comfortably. Before I dive into my speech, I wanted to start off by just saying that I think it's important not to sugarcoat things or to give the idea that something is easier than it is. So I wanted to preface my speech, or giving the idea that something's easier than it is, because from an external perspective, looking at sort of social media and things like that, you might have had the idea that us scholars have had a sort of a year without hiccups, or that kind of thing. So I just wanted to invite you all to picture a scene really quickly before I start my speech. So you're waking up from a 12-hour flight to South Carolina to Dublin after 49 hours of hurricanes, airplane malfunctions, 24 hours of queuing in airports, and you reach down under the seat in front of you to put your shoes back on after the long flight. But there's something wrong. Your shoes are filled with a liquid, they're sloshing, and confused, you turn to the lady beside you, wondering, did something spill? What happened? And the lady looks at you, at your bleary eyes, with such pity in her face, and says, "The child in the seat in front of you had an accident during the flight." So, yeah. So you arrive in Dublin, back home. You have 12 hours to pack the bags that have actually been sent to Chicago, not to Dublin, and you have 12 hours to repack those bags in Chicago and to head to Finland the next morning to start a course. So you arrive there without your bags, not all of your dive gear in Chicago, and your shoes filled with pee. At those kind of moments this year, you can't really do anything just but laugh. And now it's quite funny. At the time, it's not that funny. And I think how I got through it is by breathing. So I would like to start off my speech to both chill myself out a bit, and also I think it's good for everyone to breathe. So I would like to invite you all to breathe with me. So inhaling through nose and releasing. And one more time, inhaling through the nose and releasing. It is a beautiful thing to be connected by the breath to the world around us. From a scientific perspective, we aren't living things separated from each other. We're actually living in a soup of vibrating subatomic particles that are made of us and that are flowing in and out of us constantly. Whether we know it or not, we are connected to each other and the world around us. I asked you to take a second breath there because 50% of oxygen on Earth comes from the ocean. So whether we know it or we don't, we are deeply connected to the ocean. And I think that that's why I love the study of ecology, because it's almost a celebration of this concept. It's studying how things and ourselves are interconnected. So the goals of my year, thanks to Rolex, I was able to continue studying and learning more about this connection. And these were my kind of core 3 goals of my year. So my dive upskilling, so to improve my own diving, as I said, to be able to dive more comfortably and safely, and to be able to work underwater. Secondly, science communication, to be able to share my own research and what I've learned underwater. And thirdly, to work directly with researchers in the field to improve my own awareness and knowledge of global issues. And thanks to the Society and to Rolex, I've managed to achieve more than I thought possible this year. This quote, coined by Einstein, has repeated itself over and over in my head this year. The more I learn, the more I realize how much I don't know. Previously, I had written a speech detailing the ins and outs of every course that I've done this year, and I gave it to Maureen, and she said it was actually a bit boring, and because 30 minutes is not really enough time to delve into all of everything that I did this year in 365 days. So in short, this year I dove 254 times. I helped out or conducted 8 different research projects and attained 17 certifications. Thank you. Those including, just very, very briefly, an occupational scientific diver training with the Finnish Scientific Diving Academy. This program was set up by Ed Stockdale, who's actually here from Finland supporting me tonight, so thank you so much for coming, Ed. This course was phenomenal. It was really, really intensive, and I completely changed as a diver throughout the course. And it was the best way to kind of set me up for the rest of the year. Straight after this course, I headed back home to Ireland, and I did my commercial diver training. This training is really, really vital because in Ireland and in a lot of the UK, and in some of Europe, it's actually a legal requirement. In order to get paid to do underwater work, you have to have this cert. So yeah. As well as that, I improved my technical diving skills. So I started off with GUE fundamental training, which is kind of a precursor to technical dive training to get kind of the basic level of perfecting trim and buoyancy and things like that. And by the end of the year, actually just last week, I finished my air diluent rebreather training with Mark Powell in the UK. So it was a pretty massive milestone to hit from where I kind of started off this year. So here are the other certs that I got this year. So all of these logos that you see here, behind these logos are so many incredible people that this year have given so much time and energy and knowledge to me so freely, and for that I am eternally grateful. And yeah, needless to say, I'm a very different diver and scientist now than I was when I started out. So one of the things that was really important for me this year was science communication. So science is not finished until it is communicated. I believe that as a scientist, your role is not to become some kind of all-seeing, all-knowing expert in a certain field that reads scientific papers and only talks about it with your peers or superiors. But the purpose is to find joy in knowledge and learning and experimentation, and ultimately to raise the global wisdom of humanity. And in order to do that, you have to be able to communicate what you learn succinctly and well, so it impacts with people. So to this aim, my 3 main core scientific communication skill upskilling this year was through photography, because a picture tells 1,000 words, through report writing, and through community involvement. So rather than regale you on the sort of what I learned in terms of ISO settings and shutter speed and things like that, or to go through in detail on some of the reports that I wrote this year, I thought that I would just share with you some of the images that I took this year, and kind of nerd out a little bit with you guys, because that's kind of, yeah, stuff that I like talking about, and I thought that it would be, yeah, fun to talk about. So, yeah, let's dive in. So, this image was taken in Lembe. Yeah, he's kind of cute. This is a black-banded female, black-banded damselfish. So during 1 week of diving in this area, I observed 4 different fish of this same species exhibiting this behavior, living in very close association with glass and plastic bottles. This really interested me and kind of confused me. Why were these fish selecting this habitat over a natural habitat? Was it because it was better protection? It raised a huge number of questions in my mind. And looking closely, because after I saw more and more of this happening, I looked much closer, and you can actually see the fish were laying eggs inside the bottles. So they were using the inside of the bottles to, you can see there the kind of ribbons of the damselfish eggs. And this, again, raised so many more questions. Why are they doing this? Is this behavior isolated to this specific area in Lembe, or is it a global issue? And subsequently, since then, I've actually connected with a researcher in Spain that's looking at very similar behavior, where cat sharks are actually starting to use the mooring lines and discarded plastic rope from fishermen as in preference to natural substrates to lay their eggs on. So it's a really interesting topic, and something that's, yeah, really kind of caught my eye, and because this problem isn't always thought about, because very often the compulsion on any dive is you see rubbish. The reason why I realized that they were laying eggs, because I went to pick up one of these plastic bottles and I almost got bitten by the damselfish because she was protecting her eggs. So it raises a different question, is what happens when the things that we put into the ocean start becoming homes and start becoming used by the animals in the sea? So yeah, that was my first kind of question. Something else this year that has kind of fascinated me and intrigued me, and something that I've kind of been quite nerdy about for quite a long time, actually, were the adaptations and intricacies of nudibranchs. So this image was actually also taken in Lembe, and the reason why I love this image so much is because these are 2 dorid nudibranchs, and they're actually sexually reproducing, and you can see on the left here, this kind of big yellow structure is actually a rosette of their eggs. So it's kind of interesting. You can see the whole process happening all at once. And the reason why these nudibranchs are kind of so wild and interesting is that these individuals are what's called simultaneously hermaphroditic. So they are both male and female at the same time. And also some other really interesting features about these animals is that you can see at the back here, the sort of plume of what almost look kind of like feathers at their back is actually their gills. So they have these really intricate folded structures that increase the surface area, very similar to the alveoli in our own lungs, to be able to breathe. Also, kind of crazily, is that those gills ring their anus, so they are actually breathing the water around their backside. So, but before before you get worried about that and concerned for this species, it's natural, it's what they do. But so you know that their sense of smell is actually up front, well away from the back. So up at the front, what often get confused for their eyes, similar to in snails or slugs, they have eyes on stalks. These aren't eyes at the front. The two stalks are their rhinophores, which are similar to inverted nostrils. So they can essentially smell the chemicals in the water column. And that's how they find their way around. They do actually have little eye spots. Some of them have one. This guy has 2 little eye spots. And they can sense light and dark, but they find their way around mostly by smell. So I knew at this point you were going to be equally as obsessed with nudibranch as I am. So here's another one. And this nudibranch is also doing something really cool. It is an aeolid nudibranch, and on its back here, the spiky-looking structures are called serrata. And why I love this image particularly so much is you can see, I hope you can see, I could see just in the bottom left corner and the top corner, there are hydroids. And as I watched this nudibranch, he was, or she, I'm not sure, was eating these hydroids. The reason why this excited me so much is because hydroids are in the same family as anemones, in the phylum Cnidaria. And Cnidaria all have what's called nematocysts, or stinging cells, which are the same stinging cells that would sting you from a jellyfish. And the reason why this nudibranch is such a badass is because he's eating the nematocysts. He's able to eat the nematocysts, or she, sorry, they are able to eat the nematocysts in the hydroids and essentially circulate them through into their serrata. So the serrata, the digestive system of the nudibranch extends up into the serrata. This is just a little doodle I did for you guys. And you can see there at the bottom, that's what it looks like, what a nematocyst looks like before it's fired, and then after it's fired, they're this, even under a microscope, they're these super, super crazy looking sort of projectile. They look like something from like a medieval weapon. But yeah, these guys are just seriously awesome. So yeah, so another, okay, this is the last one, I promise. Those aeolid nudibranchs that I was discussing, they're not the only sea slug or nudibranch in the world that are able to essentially steal the powers of another organism. A lot of people, I think, know that nudibranchs look kind of like Pokemon. I think a lot of people think of them as a Pokemon of the sea, but not a lot of people seem to know that they actually have powers like Pokemon. And this guy, these are really, really awesome. So it's actually the green organisms. There's actually 4 on top of this piece of red algae here. These are a new species that were recorded, and this photo was taken in the Falklands, and this species was only documented a couple of years ago by the team of researchers that we were working with. So needless to say, I was really excited when we found these. And the reason why these guys are so cool is because they are able to do something called kleptoplasty, which is essentially they eat the chloroplasts, or the cells that are photosynthetic in algae, the cells that are able to absorb sunlight and produce food. And they can take the juvenile photosynthetic cells, incorporate them into their tissues, and they actually can become one of the only photosynthetic animal types in the world. So yeah, that's just really cool. So if you're not as equally as excited about nudibranchs as I am, I'm not sure we could be friends, because yeah, I could generally have a 30-minute slide on these, but I will save you and move on. So why we were actually in the Falklands in the first place was we were working on a scientific research project looking at the impacts of peat erosion on the benthic flora and fauna of the area. So peat is very old, compacted vegetation that has been built up over thousands and thousands of years to form basically, it kind of looks like Earth, but it's super, super compact. We have a lot of it in Ireland. I was told to describe what it is, just in case anyone needed to know. But it stores a lot of carbon. But the problem is in the Falklands that due to agricultural practices and sheep grazing, the natural tussock is these really big grasslands are being sort of wiped out. So the root structures aren't holding the peat together. And through the sun and the wind, it's drying out and blowing away, forming peat dust. And where does this peat dust go? But into the sea. And it forms this sort of mat over the seafloor and can suffocate and damage the life there. So the reason why I like this image so much is because it's of a queen or Patagonian scallop. And something that I didn't know was that scallop can have up to 200 eyes. And I like this image because you can see the kind of black circles along the rim of the scallop are its eyes. But in the center there, and also covering both sides of it, there are these really thick, heavy peat grains. So I just thought it was a cool image that kind of encapsulates what we were there to study. Also, while we were there, we saw a lot of long fin squid eggs attached to the stipes of the kelp forests. And why I thought this was quite interesting was because in the Falklands, their main economic input is actually from squid fin fisheries offshore in the deep sea around the Falkland Islands. So it was quite interesting to see that something that constitutes over 50% of this nation's GDP is being so heavily impacted by an agricultural practice on land. And it was something that the researchers were looking at and they were talking about, but was kind of new, but has obviously been going on for years. So it was really interesting to see the sort of connection between the land and what's happening underwater firsthand. Something else that's kind of fun that I didn't know was when I took this picture, I was confused. Why is this hermit crab carrying around another small hermit crab in its claw? I thought maybe it was going to eat it. I wasn't sure. I asked the researchers that we were diving with afterwards and showed them the picture, and they said that male hermit crabs apparently will very often carry around a female hermit crab, essentially waiting for her to mate with him. So I like to think that this image is of a male hermit crab bringing his wife on a walk. So there you go. Also, I thought it was really cool because you can see on its shell there are these little tube-dwelling tube worms, and yeah, I just thought it was really cool that you can kind of see this animal itself has become a habitat for other life. So something else, I knew before the start of the scholarship about the Antarctic Climate Treaty, or the Antarctic Treaty, but I didn't know that although the land in Antarctica is protected, the sea is free reign. And krill fishing there is heavily impacting the wildlife in the area. So we fish krill for omega food supplements, for feeding in fish farms, and it's also used in pet food. But it's actually the keystone species of Antarctica. Krill constitutes 96% of the calories consumed by seabirds and mammals in Antarctic waters. The reason why I like this image so much is that you can kind of see the penguins and the fishing fleet in the back, and I just thought it was an interesting juxtaposition. So, one thing that's also kind of interesting is that CAMLAR, the agency that are kind of responsible for setting fishing quotas in Antarctica, a member of who I actually met this year, would say that the fishing quotas are okay because they only allow up to a 6% take of the krill that's there in Antarctic waters. But that's actually a huge amount, and the amount of krill that is has that quota that is set has never once in history actually been collected by the fishing fleets that go down. So it's seen as this massive economic opportunity by fishing fleets. And yeah, it's quite striking and troubling. Something else that I learnt in Antarctica that is maybe a bit more fun are the scientific names for different iceberg sizes. So some of you might know this, but larger icebergs that are between 3, I think greater than 3 and 16 feet, so that's around about 5 meters, I think, above sea level, are called bergy bits. And smaller ones around the size of a kind of truck or a piano are called growlers. So I just thought that was really interesting that these kind of fun words that scientists come up with. Also, I didn't know that it's brain-numbingly cold to dive in Antarctica. Yeah, just kidding. I knew that before I went. So in Chile, I was working on a research project that was really fascinating because I was learning about the sort of connection between shallow reef systems and deep mesophotic reef systems. So I was working with a group that we did numerous different kind of scientific diving projects, but one of the ones that was most interesting and most fun was these, we were redeploying and collecting these pingers, or acoustic data collection pods? No. Pinger, we called them pingers. And they basically, the year before, marine veterinarians had collected fish from a bunch of different groups and different species and inserted these tiny, tiny acoustic sounder tags and then released the fish into the area. So these monitoring devices were put at different depths in the ocean around the kind of Chilean border. And every time a fish passed them, the acoustic tagging device would ping, and the sensor would collect tons of data about what the fish had essentially been up to, how deep it had been going, what species it was, where it was. And it was really, really interesting because every time we took up one of these tags, we learnt loads about what the fish had been up to. And is that okay? Is that me? Can you hear that? Will I just keep them? Oh. Nice. That always happens to me. I think technology has something against me, and then so I'm like, it's not working, and someone comes up, and then it works instantly. But the reason why this was so interesting, what we were learning, was because it was giving us kind of insight into where the fish were reproducing and that kind of thing, and also insight into where they were moving. So that was being able to be used by government officials in the sort of mapping out of fishing laws and where people could fish and that kind of thing. Something else that I learned this year was about the connection between again, the connection between sort of land and sea, and this is no better encapsulated in my mind than in cave systems. So on a cave diving course in Mexico, I learnt how caves are essentially like the veins of the earth. They are a circulatory system that move minerals and nutrients from one place to another. I also didn't know that most of the life, or the troglodytic cave-dwelling life in these caves, is actually descended from the ocean. So one really interesting thing that I learnt while there was a group of researchers had found a particular cave system that was full of these albino isopods, and which looked kind of like this. The top 3 there are all albino isopods, the bottom are shrimp. But they'd found a load of these albino isopods in this one cave, and it had been really interesting, and they'd wonder why, and they got ecologists in to look at it, and they couldn't figure out why, and eventually it got traced back, looking at the sort of currents and flow of the cave systems, it got traced back to this one factory, and the fact that there was wastewater being emptied into the sort of the aquifer way up inland. So it was just really interesting to see how the study of ecology can actually bring us back to land and look at our practices on land. So finally, this is a endemic Galapagos barnacle blenny. And I really like this guy. I didn't know that some small fish find these really cozy homes in ancient, old, dead barnacles in the world. And I wanted to end on this image because I think it's really important to look at the creatures in the ocean that aren't necessarily beautiful or charismatic. This blenny obviously is not in either of those groups, because those creatures can be really fascinating and interesting and can tell us so much about the world that we don't know. And they can also be vital to the ecosystem and to our lives. And we're intrinsically connected with them as well, even if we don't know it. I think that very often the sort of larger, charismatic megafauna in the ocean get a lot of the attention in the media because they're not that far removed from us evolutionarily, so we can look them in the eye and identify with them really well. So yeah, I'm really, really happy and excited to move forward, sort of championing the lives of the kind of small, funky things in the sea. So what's next? I am really excited to say that my nerding out over the small critters in the ocean is not going to end with my finishing of my scholarship. I am in September going to be starting a master's in marine science, in marine biological resources, and I'm going to get to study everything from marine genomics, marine ecology, biostatistics, and also marine policy and international governance. And in my final year, I'll have the opportunity to set up my own research project. And you can be guaranteed that there will be a lot of data collection underwater. And I'm actually very interested in looking at anthropogenic substrates in the ocean. And I think that potentially the damselfish in Lembe were kind of the ocean giving me a message in a bottle. So I would like to say a massive thank you to my family and my partner Michael, who are here tonight. I can't see you, because they are who have made me who I am today. And I'd like to say thank you to Michael, who gave me the much needed hug after I got home from that fateful flight home, and who actually brought me back to my house. I'd like to thank my mum for bringing humor to the situation and laughing her socks off when she saw me in mine. And also for subsequently washing my shoes, which I am actually now wearing. And also thank you to my dad, who got me back to the airport and who helped me hunt down those lost dive bags and got me to the airport on time to make my course in Finland. So thank you to my family. Thank you also to my coordinators, Jim Standing, Jade Berman, and Rich Somerset, who have been the driving force behind all of my learning experiences this year and who have been so incredibly supportive every step of the way and have just had my back this year. From the supportive phone calls to emails to linking me up with really interesting researchers, they really all listened to what I was interested in and connected me with experts in what I was fascinated in, and that is such a massive gift that I'm bringing forward in my life, so thank you so much. Rich is actually here right now, so thank you. And also thank you to Felix, yeah. Thank you, Rich. Jade and Jim aren't here, but thank you. Thank you also to Felix, who has been so supportive this year with my writing of the blogs. He's been my main editor behind all the blogs and has also helped to encourage me to get them all done, and in such a supportive and genuinely lovely way. And it's just, yeah, it's been awesome to get to know you too. So thank you. Thank you also to all of my hosts and mentors. I'm just completely floored this year by the kindness in the world and the fact that so many people would give so freely and so kindly of their time and knowledge. So thank you all so much for completely changing me as a diver and as a scientist. So words cannot describe how much impact you've had on my life this year. So thank you, everybody. Thank you to, I think Kirk is here, and Ed is here, and yeah, everybody else that's on this list. Thank you. Thank you also to my equipment sponsors who have kept me warm and dry and well equipped in a lot of harsh environments this year, and kept me safe. So thank you so much to all of the equipment sponsors also. Also, finally, thank you so much to Rolex for providing this opportunity, and thank you so much to the Our World Underwater Scholarship Society, all of the volunteers and everybody that is doing so much work behind the scenes. Your effort and work and everything you put into this scholarship is absolutely phenomenal, and I'm so, so grateful to you from the bottom of my heart. And I am so excited and humbled and honored to have been welcomed into this sort of family and to be kind of, part of this network moving forward. And I'm really, really excited to kind of continue that legacy and support the young divers of the future. So thank you so much for listening.
I'd like you to know that Hannah was incredibly scared about doing this. And I said to her that understand that you know more about what you're going to talk about than anybody sitting out there. And that comfort at a microphone comes from knowing your subject, and she proved that. So our returning AAUS, again, the American Academy of Underwater Sciences, Dr. Lee Summers' intern is UN Azu. Where are you? I can't see. Oh, you're coming. Okay, Marty. Welcome.
Yeah, I'm gonna need a little lower for that. All right, thank you for the introduction, Michael. Once again, my name is Yuen, and I had the pleasure of being the AAUS Summers intern. I wanted to start off by paying homage to the namesake of my internship, Dr. Lee H. Summers himself. He was a pioneer in diving safety and taught for 30 years at the University of Michigan, which is less than 2 hours from where I grew up. Unfortunately, our paths never crossed, but I'm grateful to take on the legacy of a Midwestern diving legend. I did not, however, stay in Michigan. I went over to Massachusetts to get my undergraduate degree in biology, which I just graduated this past month. Thank you. It was a long 4 years, but it was my early undergraduate discoveries that actually led me to applying for this internship. So for one, I got to explore the underwater world for the first time after getting scuba certified my freshman year. I then went on to do field work, which included diving in the Turks and Caicos Islands for a marine studies program for a semester. I also fell in love with staghorn coral during 2 summer internships with the Mote Marine Laboratory. And these experiences led me to realize that I needed to obtain scientific diving training and certification in order to dive for coral research, which is what I want to do for a career. So I'm so grateful to have been accepted for this internship, and I'm really excited to share my journey with you guys. So my host for this internship was the University of California, Davis, and my time was split up between 2 locations. I started off at the Bodega Marine Laboratory, or BML, and this is where I got to do my scientific diving training. And then I relocated to Lake Tahoe, where I was at the Tahoe Environmental Research Center to really take on my newly developed underwater research skills and truly became a scientific diver. My internship started mid-July with the first session of the Research Diving Techniques course, and this was mostly in the classroom in the pool. And one of the key things you learn about scientific diving is that the science comes second. The most important aspect is always safety, because science is never convenient. And for that reason, we spent hours on environmental assessments, risk management, emergency first aid, and rescue drills. Then we were able to dedicate some more time towards marine archaeology fundamentals and marine biological surveys. There was a bit of a time gap between the first and the second session of the marine research, or the research techniques course, and so I had a few opportunities in that downtime. This included doing a marine archaeology survey with John Harold, who was one of the instructors for the course. We went in search of a historical anchor at Duncan's Cove along the Sonoma coast. Unfortunately, we weren't able to find said anchor, but it was still a great experience putting some of those search techniques to practice. I also got to dive in the San Francisco Bay with the Golden Gate Bridge as the backdrop. And this was a really simple set of dives, retrieving, cleaning, and reattaching some underwater instruments, so very simple dives. I did similar work at the Point Reyes National Seashore. Once again, we were just doing very light maintenance work, cleaning some underwater instruments. Back at the Bodega Marine Laboratory, I got to observe a seawater intake cleaning dive. I was just taking pictures and videos of the people that were actually doing the hard work. And back at the lab itself, I got to shadow the researchers in the abalone lab, the abalone nursery, I should say. The second half of the research techniques course involved going to the Sonoma coast. So we were diving during the day in the Pacific Ocean, and at night we were camping under the stars. So it was cold, we were out in the elements, and science is never convenient. So you can see me smiling before doing my biological survey with my dive buddy, but I did not come out of that water smiling because my hands were numb and I was really, really cold, not a happy camper. On this dive, which was our very last dive, we were actually supposed to go see a shipwreck, the SS Pomona, but the visibility was just so bad that once we got out there, the instructors couldn't even find the shipwreck themselves. So we turned it into a navigational dive on the spot. And I can say I came out of that dive smiling because my dive buddy and I made it back directly at our entrance point. So it was a confidence boost moment there. Thank you. Back at the campsite, we did have some fun bringing back and dissecting some sea urchins, which you could see right there with my friends and my instructors. I actually found out then that the parts that we like to eat are the reproductive parts of the urchin, so that is a fun fact. We also had an OWUSS cameo, so Abby Diaz, who was the 2019 Dan Diver Safety Education intern, came to do a presentation on marine photography. So it was really great to see her and also her beautiful photos. So there were many tangible and memorable accomplishments I made during this course. I got a lot of certifications, which included the AAUS Scientific Diver certification, which, you know, that was the point of my internship. But I also had a lot of other experience, Pacific Coastal Marine Species Identification, marine archaeology, cold water diving experience, which is actually a big one for me because I'm kind of a baby. I've mostly been diving in warm water, so I was not used to wearing so much neoprene and having less mobility. So I really actually struggled diving for the first part, but eventually I was able to get my gloves on by myself. So I was actually really proud of myself. I also made plenty of friends and memories. My fellow teammates or course mates were amazing people, and also the instructors were awesome. For the second half of my internship, I relocated to Lake Tahoe, and this time I was working with the Tahoe Environmental Research Center field team, and we had about 2 weeks worth of field dives to do, and our task was to do a survey of the invasive Asian clam population in Sand Harbor, which is on like the northeastern shore of Lake Tahoe. So these invasive clams are causing an aesthetic issue of Lake Tahoe, which is a huge deal because Lake Tahoe is really known for its like clear blue waters and its picturesque shorelines. And so these clams are causing unwanted algae growth because their excrement is accumulating and becoming a nutrient source for huge algae blooms to grow and cloud up the water, wash up to shore, does not look very pretty. So our dives involved taking little minnow traps and sifting out sand to count the number of clams on predetermined transects. And we also did excavation plots where we put down a meter by meter quadrat and collected all the clams within that quadrat, which we preserved, brought back to the lab, and measured the size of every single clam. And you can see me looking really cute in my lab attire with a caliper in my hand. So this sizing data is really important because the size of the clams determines their sexual maturity, and reproduction is a really important aspect of population growth. The overarching goals of the project was to do a three-year survey of the population at Sand Harbor, and then after those 3 years, create a successful mitigation plan in order to limit the numbers of these clams in Sand Harbor. So I have these beautiful figures to explain, but first I wanted to say that these are all credited towards the Turk field team. My internship ended before they were able to do data analysis, so all credit goes to them and not me. The first figure shows a heat map from our transect data, and you can see that there is a huge blob that indicates a very high concentration of the invasive clams in the northern shore of Sand Harbor near the boat ramp. And this data alone is a really good indicator for the mitigation program to say, oh, this is a really good area to target in order to reduce the numbers of the clams. Next up, we have the data from our excavation plots, which shows the various size classes of our clams. So the white dots indicate the plots where all of the clams were juveniles, and the progressively darker pink dots indicate the plots where there were higher proportions of adult clams, so those that were sexually reproducing. And so this data is important because looking at the first year's data, which was 2021, and comparing that to 2022, there's actually a lot more juveniles in Sand Harbor. And this is showing that there's increased reproductive rates of those adult clams, which is very concerning because we want to be limiting their growth, not letting them grow, or populate, I should say. Lastly, I wanted to show a direct comparison of the first and second years of the survey, and this is showing several colors. The tan dots and lines are representing where the clams are found in both years, and the yellow is showing where they were found only in 2021, and the purple is where they were found only in 2022. So we can see that they're mostly found in the same areas from both years of the study, but there's one area of concern that's highlighted in that in that red area up in the North Shore, and once again, this is showing area for concern because we can see that they are spreading in the northern shore of the boat ramp. So in order to sum up my experiences of the clam surveys, I have to say I had an amazing, memorable experience. The dive team was awesome. They were really good mentors, and I couldn't have had a better time as a novice AAUS diver among them. The days were long. We did get smoked out by the California wildfires for a couple days, and I got swimmer's ear for the first time. But what can you say? Science is never convenient. And all in all, I just had an amazing time in Lake Tahoe, and I was really, really sad when the clam surveys were over, actually. However, my internship wasn't completely done yet. The last week of my internship was reserved for more training, and this time I was able to get dry suit and altitude certification. And I have to say, I looked really good in a hot pink dry suit. But in all seriousness, it was another great opportunity to get more tools into my toolbox, and it was the perfect ending to my internship. It's been almost a year since my internship started last year, and I have to say I would not be where I'm headed now if it weren't for this internship experience. I'm really grateful to say I got my dream position at Biscayne National Park, where I'll be working as an ecology assistant for a year, and I'll be doing a lot of coral restoration work. After that, it's hopefully on to graduate school. I haven't decided whether I'm headed towards a master's or a PhD yet. That's a future issue, I should say. Not an issue, but a future thing to settle on. After that, I'm hopefully going to be doing coral reef research and restoration work. There are a lot of people that I need to thank for making my internship possible and such a success. Of course, all of the volunteers at the Our World Underwater Scholarship Society. I'd also like to thank the AAUS Foundation and the California State Parks team, as well as all of the folks at the University of California, Davis, including those at the Bodega Marine Lab and the Tahoe Environmental Research Center. So thank you all. Last but not least, I'd like to thank Dr. Lee H. Summers himself. You have changed my life. Thank you very much.
So, going back to that last photo that Ewan had up on Lee Summers, just on a personal note, I call them the three wise men. It was Dr. Len Ekstrom, Dr. Lee Summers, and Dr. Art Bacharach. And when I did not have degrees in marine science or medicine, but those three men, I was an investigator, a researcher, and those three men took what I did and gave me a platform to bring that to the diving community. And I tell you that because all of our scholars and interns are building that Rolodex. Young people don't know what a Rolodex is, it's okay. Building that Rolodex of mentors, of supporters, of colleagues. And that is the most valuable thing in science and research and life, I think. So one of the things that we do as a society is allow them to build that Rolodex, and it makes careers. So on that happy note, our returning intern for the Divers Alert Network Safety Internship, Aaron Kissel. Aaron, I can see.
Hi, everyone. Thank you for coming. I know it's been a long morning, so appreciate you listening. My name is Aaron, and I was Dan's diver safety intern last year. And although normally it's the diver, Dan safety and health intern, we do also focus on health, even without the title. So a little bit about me. I am originally from Southern California, grew up diving in Catalina, and I learned actually dive at the Boy Scout camp called Emerald Bay. But I didn't get involved into hyperbaric medicine until I met Carl Huggins here in the audience, and then started volunteering at the Catalina Hyperbaric Chamber. When I went off to college, I studied economics, which is probably a bit unusual for the society. But I'm also taking pre-med classes and plan on enrolling in medical school next year, assuming that applications go well. We'll see in a few months. And then a little introduction to Dan as well. I'm sure most of you know at this point, but Dan is a nonprofit, and we serve divers all over the globe. However, most of our members are in the United States. And Dan has a bunch of different departments. The insurance is probably the best known, but there's also the marketing department, the research department that Alex talked about, and then the two that I was split between, which are the medical department and the safety services department. And I did half my week at each, so I was there for 12 weeks and did like 2.5 days of each week. And then I was one of 5 interns as well. So next to me in the photo here is Liam, who I worked with every day, and we sat next to each other. And then across the way are Gabe, Alex, and Beth. Okay, so to start with the Safety Services Department, it's led by Francois Berman, who may be a familiar name for some of you. And they have a variety of different projects, both in the water and out of the water, focusing on dive safety, risk mitigation, and hyperbaric medicine. And for those of you who don't know, a hyperbaric chamber is used to treat patients with decompression sickness. So after you get out of the water, if you have symptoms of DCS, you'll go into the hyperbaric chamber, and you'll be in there for hours at a time, sometimes multiple treatments before coming out, and hopefully those symptoms resolve. Some of the projects I was involved with dealt with risk mitigation out of the water, so we were making protocols for different dive operators to reduce the risk that they have of accidents. But more importantly, we were doing actual hands-on training. So on the left there, you can see a aluminum scuba cylinder, and that's the top, that's the neck there, and we were inspecting that for the integrity of the metal inside of there. And then on the right is a scuba valve that we were taking apart and putting back together to check the O-ring integrity, and then the other components as well, trying to make sure that we learn the proper way to do it. So at the Safety Services Department, I had one main project, and that was to build a new manual for training chamber operators how to use a hyperbaric chamber. So with the hyperbaric chamber, there are three main positions. You have the tender who goes inside with the patient, the recorder who does all the timing and then sets the schedule, and you have an operator who actually turns the dials that dive the chamber and then bring it back up. So there's various aspects that the operator needs to know about the engineering of the chamber and maintaining the patient safety. But most importantly, I was taking a bunch of different materials from fragmented manuals and putting them into one more comprehensive one. Some of those materials came from Dan USA, Dan South Africa, Catalina Hyperbaric Chamber, and then some independent research that I did myself as well. And there were also some newer things that were added. So this was done during the COVID-19 pandemic, and while there hasn't been that much research done on COVID-19 in a hyperbaric environment, there are a few things that we also do need to know. So in a hyperbaric chamber, you have denser air. And with that denser air, if you were to cough, it's going to take longer for that air to reach the ground. So that could mean that COVID-19 could stay in the air longer than it normally would just here in an auditorium or anywhere else in the world. So as we're making protocols, we were thinking of things like, how do we make sure that in this enclosed environment, somebody can stay safe if that patient were to have COVID-19, and the other various aspects like the humidity and the fire safety as well inside of a chamber. And then the eventual goal of this manual is to have it transition to an e-learning course. Some of you may have taken Dan's other courses. And then that would be translated into a bunch of different languages. Most importantly, right now, we're working with Indonesia to try and set up more hyperbaric chambers there. And then moving on to the second half of my week, I was in the medical department, which was headed by Dr. Nochetto and Dr. Chimiak, also working at Dan as physicians. And the medical department, you may know from the Dan hotline. So if you've ever had a dive accident, you can call Dan either on the emergency or the non-emergency hotline, and somebody will pick up, usually one of their diver medics or a physician, and then you can get emergency response wherever you are. But what also happens is they collect data from each of those cases, and then we can use that data for research, which is what I was doing. So when I arrived at Dan, they were focused on something called inner ear decompression sickness, which is just the bends of the inner ear. And we were looking at previous cases that had happened and trying to analyze patterns of that data. So on the left here is the Gulf Chamber, which is at Duke Center for Hyperbaric Medicine. And that's a chamber that can go to well over 1,000 feet equivalent, and they study things like high pressure nervous system and other technical diving ailments. And then in the center there were all the books that were handed on our desk on the very first day that we arrived, I think within the first 5 minutes. And we were reading those for the first 2 weeks pretty intently so that Liam and I could get up to speed on all the physiology that we needed to know. And then on the right there is the proposal that we sent to the IRBs to get our study approved. And the study that we're conducting, we started last summer during the internship, but we're actually still doing it today. And Liam and I are both working remotely to get that done for Dan. And then to talk about IEDCS, so it's something that I hope none of you have had in this audience, maybe I'm wrong, but it's something fairly rare in recreational divers and traditionally scribed in technical divers. And the reason for that is it occurs often during gas switching. So on the left here, you can see this graph that shows what can happen if you're gas switching. And in the blue line there, you're off-gassing off your old gas, and then you're switching to this new gas. And if you on-gas with the new gas faster than you off-gas with the old gas, you can actually stay at the same depth and end up with decompression sickness because the cumulative effect of that is this red line here. And that's what we were analyzing, along with other things for recreational divers. And then in the center here is a diagram of the inner ear. So up at the top, those are those three rings that are your vestibular spaces. And if you were to have gas buildup in that area, you would expect to have loss of balance, dizziness, and vertigo, which are the most common symptoms of IEDCS. And then down at the bottom here is the cochlea, that snail-looking shape. And if you were to have issues there, you'd expect problems with hearing. So another thing that can happen with our patients is they would have loss of hearing or ringing in the ears that can last for months or actually for a lifetime as well. And then on the right here shows a delay in time that can occur with IEDCS. There's a lag period. So often as we're looking at patients, we would see that their symptoms wouldn't occur until 12 or sometimes even 18 hours after they got out of the water, which can sometimes be a problem because if they were to leave a location that's close to a hyperbaric chamber, it could then make it very hard for them to get back to a chamber and seek treatment. And we also had time to do some field trips while we were at Dan. It's located in what's known as the Research Triangle down in North Carolina. And on the right here is Duke's Center for Hyperbaric Medicine, which is by far the most complex hyperbaric facility I've ever seen. They have chambers that are the size of this room. They have some that are small enough you wouldn't fit in them, and everything between. They also have MDs, PhDs, chamber techs, and there's just tons of people running around that facility all the time. And then they're also doing both research and clinical medicine treating patients. And then on the left here is an air compressor from Bauer Compressors. And while we were looking at the manufacturing process, we were also watching the air filtration systems being built and maintained. And I think, as many of you probably know as divers, if you ever were to have air that smells bad in your scuba tank, you immediately want to get rid of it, and anything that came from the same facility, as likely it's been contaminated or that air filter isn't working anymore. And one interesting aspect that we were doing in safety services is Francois Leader actually visited California during the wildfires and tested the air filtration systems in places where the skies were so smoky you couldn't even see clouds or anything like that. But the air filtration systems were actually good enough that you could still dive on that air, and it was perfectly safe to do so. And then I think coming into this internship, I was probably one of the lesser experienced divers. I'd been diving now for 10 years, but I only had my rescue certification. So while I was at Dan, I got some other certifications. I got my nitrox diver certification. I got my Dan DFA Pro, which is related to first aid administration. And then I also got my oxygen cylinder cleaning, valve repair, and visual cylinder inspector technicians. And all those I may or may not use in my professional career as a physician, but I think they're really good to know as a recreational diver. So if I walk onto a dive boat, I can immediately know, hey, this is a safe tank to use, it's been inspected, it's been certified. Or more importantly, if I walk into a boat and I see a lot of red flags, I can immediately know, okay, I shouldn't go on this dive, and the people on this boat also probably should not go on this dive. And then as Alex mentioned, there were some quarries that we could go around Durham and dive. So the two here, on the right we have Fantasy, which Alex showed the visibility in there, which was not the best. And then the left here is Fantasy Quarry, which was somewhat better. And there were some fun things, like they actually had a basketball court inside a lake that you could go play at. And while I wasn't doing research at this necessarily, we were doing some skills training and some fun dives on the weekends as well. And then I'm a fairly outdoorsy person, and I was able to do a lot of the activities that I loved doing while I was in Durham. So on the left there was a hiking trail out on the coast of North Carolina. And then in the center here is a bike trail that was actually only 5-10 minutes from Dan's headquarters. So after work most days, when I'd had my head on screens and in books too much, I would take some time and go mountain biking. And then on the right here is a picture of the Outer Banks, which was a great surf spot, and I went there one weekend. So I think this internship turned out to be an awesome way to do some of the academic subjects that I love during the day, and then also just explore a new place after work and on the weekends as well. And for that, thank you to everyone who made it possible, both at OWSS and Divers Alert Network, and along with support from everyone else. And for those of you listening, thank you for being here.
Aaron, I'm sure that a number of people here have the same question. If there's a basketball court at the bottom of a quarry, and basketballs are generally filled with air, how do you play basketball?
I don't know what it was. I think it was
a rock or something. It was a bowling ball. Okay, we have to deal with that. Thank you very much. Our final presenter, yay, we're going to bring home the North American scholar, Rosie Poirier-Mosen. Where are you, Rosie?
Thank you, Mike, for the kind introduction. So as he said, my name is Rosie Poirier-Mozoon, and I am the 2022 North American Our World Underwater Scholar. And today I'm presenting to you Oceans of Hope, which is a year of search of ocean solutions and community science. So this is me standing here on the same stage 1 year ago and thinking deeply about how I wanted to approach my scholarship year with intention. And at the end of the day, I decided to focus on the intersection of humans and the oceans. So we as humans rely on the oceans for so many key services, and they typically fall under 3 different categories. Provisioning services, which include food, minerals, fuel, transportation. Regulating services that include oxygen, stabilizing our weather patterns, storm barriers, providing storm barriers. And then the third, which is often overlooked but equally important, are cultural services. So the way that we learn and play and interact with our oceans, the things that make us intrinsically human, scuba diving, surfing, all those things fall under that category. So we rely so heavily on the services that the oceans provide for us, and yet the oceans have been struggling. Because with all good relationships, there has to be a healthy give and take for it to be sustained for the long term. And we have been taking far more than we've been giving, and has been disrupting this balance. And my question for the year was, how do we plan to change this moving forward? So I decided to focus on 4 primary pillars, and that included first management, which was fisheries and MPAs, and then science, citizen science, eDNA, cutting edge science, storytelling with film and photography, and the last was culture, our interactions, our economies, and the way that we want to adapt for the future. So my first scholarship expedition took me to the west coast of Canada, a place that I'm rather familiar with, to study ground fisheries management aboard a halibut fishing boat for 2 weeks. And I was really, really excited about this because when I studied biology in university, I really wanted to take fisheries management courses, but there were none offered at my school. And when I asked my professor, he told me in very clear terms that the reason why they weren't offered is because fisheries science was garbage, and there was no such thing as good fisheries management. And I thought this was really interesting because we live in a world where 1 billion people on this earth rely on fish for their primary source of protein. So to just disregard it in that manner shows a lack of commitment to effective management and finding solutions. So with all things that you're told not to learn about, I just became ever more curious. And I had built up approximately 6 years of questions by the time I arrived on this boat. So I was really excited, and thankfully we were at sea with no internet service, so I had plenty of time to ask. So I got to join Tiare Boys and her amazing fishing family, and they involved me in all aspects of fishing. I just dove right in, and while we were working with our hands and pulling up fish on the boat, I would ask them my questions, and they were Tiare and her father both had master's degrees, and they shared with me all that they knew. And I was so excited to learn about this. And one of the really interesting things that I learned was about the quota share management that is unique to Canada ground fisheries management. And a lot of other countries have come to learn about this system. Because with quota share, they account for bycatch within the quota. So that would mean that somebody holding a halibut fishing license is also given a certain allocated quota for other fish that they're likely to pull up alongside. And that takes into account the fact that bycatch is often inevitable. So then also in the same way, halibut, when the quota is put out, let's say 70% of it goes toward halibut license holders, and the remaining 30% will be allocated for quota for other fisheries that are likely to pull up halibut. So then that way, instead of all the fishermen pulling up each other's fish and then throwing them back in the ocean because they don't have the allowance for it, they're allowed to keep a certain amount, and it creates less waste within the system and is far more effective. So after having that chance to dive deep into fisheries, I wanted to learn about another form of research that plays into food security as well. So I went to the Moss Landing Marine Laboratories and got a window into aquaculture management and fisheries science there. So here in this image, you see this rather slimy looking fish. It is actually quite interesting. It's called a prickleback, and it has the potential to be able to reduce the carbon footprint of the unagi market. So unagi is quite popular in the sushi market, and it's a freshwater eel. And it's also a carnivorous fish species, which means that it relies on a lot more resources to be able to raise it. Whereas the prickleback is a herbivorous fish that has very similar texture and taste to unagi. So what they were researching on the Moss Landing was how to grow the whole life cycle on land to see if there's the potential to be able to effectively replace freshwater eel with prickleback, and thus reducing the carbon footprint of unagi on the sushi market. So everything that they were doing at Moss Landing was in this manner. They were trying to answer relevant questions around food security and fisheries and economics. And it was really exciting to see science with such an applicable approach. So at this point, I was about 2 months into my scholarship year, and I had not yet hit the water. So I was really eager to get to dive deep with Kurt Kroc in the Cayman Islands, alongside Millie and Hannah, the two other scholars, where we got to do our freediving training alongside one of the best. So we spent our first few days learning about the human body's ability to adapt to breath hold diving, and learning about certain things about the human physiology, such as the mammalian reflex, which means that you are able to hold your breath longer when submerged in water than if you're trying to hold it on land, due to the glands. I'm not sure if I'm explaining it correctly and scientifically, but your skin recognizes the water, and you're automatically able to hold your breath for longer. So by the end of the static days, we were holding our breath for over 4 minutes. During this time, we also had the chance to go to the Cayman Turtle Center, which is just up the road from where we were staying. And I was really excited about this because they had a really unique approach to combining food systems and conservation. So I didn't know this, but in Cayman, it's actually it's part of the culture to eat sea turtle. And locals were harvesting sea turtles for many, many years, and then eventually sea turtle populations were declining due to wild harvest, but also due to the environmental challenges that they've been facing. And so the government put a hard ban on harvesting sea turtles in the wild. But recognizing that this ban would also likely result in a lot of poaching and a lack of compliance, they developed the sea turtle farm. So they have a certain amount of green sea turtles that are at the farm, and they nest on a beach that's fabricated on the farm. And a certain amount of those eggs are raised, and then they're sold as a sea turtle meat, and the remainder are part of the hatchling release program. And since the implementation of this program, they've been conducting research using DNA and extracting DNA from the sea turtles that return to nest, and have been able to determine that 80% of the sea turtles returning to nest in Cayman have DNA linked to the farm turtles. So it's been hugely successful, and they've seen a really large rebound of sea turtles around the islands. So I thought this was such a fascinating approach because they were able to combine food security and food systems with conservation in a really unique manner. And so I had the opportunity to deep dive into food security and fisheries management, and I wanted to focus the next part of my year on marine protected areas, as they go hand in hand with fisheries management. It's really important to conserve key habitats, such as nursery grounds, migratory pathways, areas of high biodiversity, because without those preserved areas, the oceans cannot be sustained, the ecosystems cannot be sustained in a healthy manner. So there was no better place to go and explore this topic than the Coral Triangle region, which is one of the most biodiverse regions of the Pacific Ocean. So there I got to work with 2 different organizations. One was the Coastal Conservation and Education Foundation in the Philippines, and the other one was the Coral Triangle Center in Bali in Indonesia. So with the Coastal Conservation and Education Foundation, I got to focus primarily on learning about the process of establishing and maintaining MPAs, as well as community collaboration. So CCEF had worked very closely with the local fishing community in order to develop their programs. And at first, they had a lot of hesitancy from the fishermen because they thought that management would mean that they're not able to fish and they're not able to support their well-being. But as they started to work alongside CCEF and see the benefit of the program and see the management measures implemented and then see a rebound of fish, they became very supportive of the programs. So at the moment, it's concentrated in the region of Cebu, but they're trying to expand throughout the Philippines due to the large success of these programs. Next, I went to Nusa Penida Island to join CTC at one of their marine protected area project sites. And here, I got to focus more on the monitoring of marine protected areas. How do we collect data to ensure that the marine protected areas are having the results that we're intending to see, to be able to collect data on the biodiversity, and the benthic community. So during our time in Nusa Penida, we were doing 2 different methods of data collection. The first was point intercept transect, which requires a trained individual to swim along a transect line and collect data at regular intervals along the transect. And they have to be able to accurately ID the different species of coral and the different species of fish. The second method that we were trialing for the first time there is called image-based photo quadrat. And it requires less training because the individual doesn't need to be able to ID the different species. They just have to be able to dive and then use this contraption here that is set along the reef bottom and is able to take photos at regular, and they're able to take photos at regular intervals. And then those images are then uploaded to an image database, and AI software is used to identify the different species of coral. So it opens up the door for a lot more community involvement in data collection. And hopefully, if it is successful, we'll be able to provide opportunities for community members to engage in science. So we were trialing the 2 methods side by side in order to see, to compare their accuracy of the 2 and see if the image-based photo quadrat method could be implemented in the future. So having this experience with community science, I next got to go to Raja Ampat in Indonesia, which is a remote collection of islands and villages in the northeast part of Indonesia, to work with Gita Anastasia and her island community. So Gita is a dive shop owner and the director of Mora, which is it's led by Indonesian women for Indonesian women. And their aim is to open up opportunities for island women to learn about ecotourism, about marine conservation, and build capacity in these fields. So in the top photo, that's me with all of the executive members of Mora, and they come from all different backgrounds. Some of them work for government, some of them work in the environmental sphere, some of them are business owners, and they have all experienced firsthand the challenges of being able to take these positions in their field in Raja Ampat, and they want to make it easier for other women to be able to do similar things. So they're dedicated to providing educational programming and dive training for island women from their communities so that they can make it easier for the future generation to have access to the same opportunities. So I was really, really inspired by the work that they were doing and was so excited to see community science in action. So at this point, I had been focusing a lot on fisheries and MPA management and was really curious about the science behind management. Because in order to be able to manage our resources effectively, you have to collect enormous amounts of data to be able to observe trends and be able to determine what policy is needed in order to conserve these marine environments. So the next part of my scholarship year took me to La Paz in Mexico to join the Applied Genomics Lab with Adrián Manga Vega. I'm going to go back to this slide. I was really lucky that Phoebe did a great job of explaining eDNA, so I don't have to go as in depth. But essentially, eDNA has great potential in helping us to collect more accurate data on biodiversity within our oceans. So typically, in order to collect data on biodiversity, we either have to use extremely extractive methods, such as bottom trawling or some kind of fishing net and pull up the different species and count them, or you have to dive into these environments and visually survey the different fish, which is challenging because some species of fish are not easy to see or maybe are only nocturnal. So there's a lot of bias in these methods, whereas eDNA is a much less biased method and also helps with the process of collecting that data. Because, for example, if a survey site is very hard to reach, it's very deep, it has a lot of surge, you can only dive it at certain times of the year, it can be really challenging to collect data. But with eDNA, all that you need is a water sample. So they can collect water samples from the surface, or they have contraptions to be able to collect these same water samples at depth. And then from that water sample, the DNA tissue is analyzed, and then you can determine the biodiversity of that area. So I was really excited about this, and there's a lot of potential for eDNA in the future. So there's a potential that we'll be able to develop technology to determine population sizes and biomass in the future. But at the moment, what it's accurately able to do is analyze biodiversity. So this is me with a whale vertebrae on the beach in La Paz. We just came across it because there are a lot of gray whales in this area. My next opportunity to learn about innovative research techniques was with the Miaru program in Fumula in the Maldives, alongside former scholar Zuzu Askin, who was last year's European Scholar. And with them, I got to see a lot of the methods that they were using for data collection. So this is a recently founded nonprofit, has just in its first year of field science, but they've already been incorporating some amazing technologies. So they have a collaborative project with Nat Geo, where they're sending deep sea cameras down to 3,000 meters to be able to determine shark populations at depth. And then they're going to be integrating AI camera at the famous shark site just outside of the Fumula harbor. And if that camera is able to be positioned at the right spot, it can record the different sharks that are coming through and identify the different individuals, which will help with the population study of the resident tiger sharks. And if they're able to get funding for a second camera, then it can be placed in proximity to the first, and the data can be triangulated, and we can even determine things such as growth pattern of the sharks. So I was really excited to see this kind of science and see the directions that technology can take us in the future. So that's me with the Miaru program, and you can see Zuzu on the far left. I had to think about that. So up until this point, I had been focusing on things on a very local level. So learning about local management, community programs, and I was starting to ask myself, okay, but how do we change things on an international scale? Because the problems that our world are facing are large and require an international push in order to effectively address them. So this led me to attend COP27 in Sharm el-Sheikh in Egypt. And this is the international climate change conference that happens once a year. And here I got to engage in discussions around oceans and ocean health. I got to attend the Ocean X Climate Summit, just a full day just about ocean presentations and projects that are happening around the world. And despite these encouraging events, the overall feeling that I had from COP was actually very discouraging, because there is a lot of lobbying that happens at COP. There's a lot of corporations that come to be able to push against sustainable progression, and a lot of energy put towards things that don't feel like they're actually making change. So I was feeling pretty heavy, and on the day that I was feeling the heaviest, I was chatting to the person next to me on the lunch line, who turned out to be Dr. Colin Young, who is the executive director of the Caribbean Community Climate Change Center. And I was chatting to him, and he was telling me that he's been coming to COP since the very first one. It's now at COP 27, so he's been going to it for 27 years. And in my discouraged state, I was like, how do you find the energy to keep coming back? Like, this is really hard. When the progress is so slow, the problems are so big, and it feels like nobody's making any change. And he said, well, you know, it is hard. But COP is the best mechanism that we have for global action on climate change. So if the people who don't care don't keep showing up, then the only people who will be here are the lobbyists and those with their own interests at heart. So we have to keep showing up, even when it's hard. And he was right, and I needed to hear that in that moment. Because the truth is, the best way to give power to the oppressor is to believe that nothing can ever change and to not even make an effort. So we have to keep showing up, even when it's hard. But that brought me to the question of how do you inspire people? How do you make them feel like change is possible? And this took me to the next step of my scholarship year, which was storytelling at the BBC alongside Yali Bosinger, who's a former Australasian scholar in Bristol in the UK. And storytelling is an extremely effective tool for change. You can inspire people, you're able to share information, and I wanted to learn more about it, so no better place than to do it here. And I got to join them at the beginnings of Blue Planet 3 when they were at the story building stage. And I got to work with the editors and the producers and talk about story beats and the elements of a story that make it intriguing, that keep someone hooked, and that make them inspired. And this was a really important time for me because this was about midway through my scholarship year as I was starting to think about how I wanted to shape my end-of-year film. What was the story that I wanted to tell? And what are the stories that I want to tell in the future as well? But with all storytelling comes the need for technical skills, and those include camera skills as well as diving skills, and I wanted to dive deeper into these elements during my scholarship year. So I had the great opportunity to do my underwater macro photography training with Reef Photo and Video at the Blue Heron Bridge, which is a famous macro dive site. And here I was learning to shoot with my strobes for the first time. So up until this point, I had done primarily natural light photography, and I was quite comfortable with that. But I hadn't really worked with strobes, and strobes are a whole different story. They're a lot more technical. You have to understand lighting, and my first few pictures were absolutely terrible, and I didn't include them in this slideshow because I'm quite embarrassed by them. But they were overlit, overexposed, red, loads of backscatter, and I was doing so terribly, and I was seeing these amazing creatures. Like, I was seeing a seahorse for the first time in my life, and I was just trying to get a good picture of it, and all I was getting was this backscatter garbage. And so, even though I told myself, I'm here on this workshop to learn about my strobes, I was starting to get really frustrated. So I turned my strobes away and had them pointing at nothing, and started just taking photos with just natural light. And Tanya Burnett, who's one of the instructors, swam over, and she sees my strobes pointing nowhere, and she's like, "What are you doing?" And she sits with me and she turns them back, and I take a few pictures and she looks at them, we adjust the settings, and bit by bit I was starting to take photos that I liked. And so by the end of the 3-day workshop, I was feeling ready to join the Atlantis Image Makers in the Philippines, who are a renowned group of underwater photographers, ocean creatives, led by Marty Snyderman, who every year go and spend 2 weeks in the Philippines taking photos together, sharing creative ideas as well as creative techniques. And these photographers were extremely generous with their knowledge and set up a few workshops and to be able to knowledge share. So this is Richard Salas on the right. I promise I know my rights and lefts. And he's doing a lighting workshop for us and teaching about the different forms of lighting that you can do for macro. And then Lenny Rosenfeld on the left, who's teaching about color grading in film. Did I say it wrong? Oh, but it's right and left for me. So I'm on the stage, so it gets to be my way. And we were seeing so many amazing critters, and by the end of this, I was starting to take pictures that I was quite proud of. So you can see there a pygmy seahorse and a couple of eels on a night dive. So now I had gotten my underwater photography under my belt, but I wanted to dive deeper into technical dive training, which is something I had never tried before. So I got to do my scientific diving course with OWUSS, followed by my rebreather course in Florida with Jennifer Mayfield, who is an instructor on the AP Inspiration. And rebreather is quite challenging. And I knew it was going to be a challenge, but I didn't quite realize how much. Because up until this point, I had been diving for more than 10 years, and I was really comfortable with my buoyancy. And certain things that I felt at ease with, I felt like I was relearning all over again with my rebreather training, because everything is different. And you don't just get to learn one skill at a time, you have to manage all of them simultaneously. And rebreather training requires a lot of emergency drills and learning how to manually control your oxygen so that you're not entirely reliant on the computer, so that you know what to do if you have any kind of equipment failure. So I was really struggling. And I think often with the scholarship, as a scholar, you feel like you're meant to just go into these things and just be good at them. But the truth is there's often a really, really steep learning curve. And so we had done the pool drills, and it was our first day in the ocean, and I was struggling hard. So we're doing these emergency drills, and my buoyancy was just terrible, and I was floating up, and I did the one thing as a diver that you're never meant to do, which is swim down with your feet up, and especially with a rebreather. So I'm like kicking my way back down, and I finally get to the bottom, and I just grab hold of a rock, and I'm holding onto that rock, and I'm just breathing. And Jennifer swims over to me, and she's like, "Are you okay?" And I was like, "Yeah, I'm okay." Meanwhile, my mask is like filling with my tears, and I'm thinking to myself, like, maybe this isn't for me. Maybe I just I'm not a good rebreather diver. And just as that thought is going through my head, this beautiful hammerhead shark, a 10-foot hammerhead, great hammerhead, swam right past the two of us, and then swam right away again. And it felt like one of those signs from the ocean, in that moment that you really need it, that you're going to be okay and that you can do this. And things got easier after that point. Here's a picture of me struggling to get into my gear. It's quite heavy. And I was really grateful for this training and the timing of it, because I had taken about a month break from my scholarship at this point, and was just easing back into it and was having a hard time getting back into the rhythm of diving and to find the joy in diving, something that I had always enjoyed. And Jennifer helped draw that back out of me. She was an amazing instructor, and she was really efficient with the skills and with her teaching, but she was also a really great person. And we had a lot of fun during our dives and during the safety stops. With a rebreather, you don't make bubbles, so it's a lot easier to hear each other's voices. So on the safety stops, we would sing the tune of songs and then try and guess them back and forth. And it really helped bring that joy of life back to me at that time. So the next thing that I got to do, which is also quite technical, was cave training with Brian Kekuk in Abaco in the Bahamas. Brian is an amazing instructor and a fantastic steward for these cave systems. And I was learning to do my side mount configuration for the first time, and learning all the different elements of cave diving, which along the same line as rebreather training requires a lot of safety drills. And the most important thing in cave diving is your line, because it really is your lifeline. So the majority of the drills centered around maintaining your line, maintaining contact with the line, setting the line. But during all of this training, we also got to explore the incredible landscapes of these caves. So in the middle picture there, you can see me, I'm shining my dive light under a salt stone. So similar to the ones that you would find in people's homes that they have as lamps, these are the same type of stone. And they glow when you shine your light through them. And I was so mesmerized by these caves, and I had no idea that I would feel that way, because in my mind, I was used to these ocean ecosystems that are full of fish life and extremely biodiverse, whereas within the caves, you don't really have a lot of living life. But instead, you have this amazing history that's written within the walls. So this stone is red like that because of the deposits of Saharan dust that have blown across the Atlantic Ocean and settled within these caves during the periods when they were dry. And we were finding clay sediments all over the place, and Brian would reach down into what looked like muck to me and then pulled out a crocodile jawbone and all these things that were within the caves, all these different formations. Each cave looked very unique, one from the other, formed by different geological time periods. And it was incredible to see this. And what I realized is that the earth has been writing its history all along. It's just us that's been learning to read it. And there it was, just on the walls of the cave, and I was mesmerized. And one of the things that forms these structures within the cave are the different water levels. And these are influenced by the glacial processes of our world. So certain caves were formed at by different water levels, and you would have areas that were just stalagmites and stalactites, and others that were formed from water flow and didn't have any crystal formations whatsoever. And it was amazing to see this. So the last experience that I'm going to share with you comes to the core of those water levels. It's the land of the glaciers and ice in Antarctica, where I got to join blue-green expeditions for a 2-week-long dive exploration trip. So here I am with Faith Ortiz, the founder of Blue Green Expeditions, as well as Vinny Malkowski, our well-loved OWUSS now step-down chair member. But it was a fantastic trip. I learned so much. The staff aboard Blue Green Expeditions provided a lot of amazing educational opportunities for us to learn about the geology, the biology of the Antarctic landscape. And the polar regions really are balancing the climate as we know it, and they play a huge role in the ecosystems that we know around the world. Even though they're remote and far away, they're influencing the world as we know it. And very few people ever have the chance to travel there, let alone to dive there. So I felt really honored to have this opportunity. And with that also comes a responsibility for stewardship to ensure that this is an ecosystem that can be explored by people in the future, that it doesn't disappear. So one of the most impactful moments from this trip was our very last dive, where we had the opportunity to dive with whale bones at an old whaling station. And this dive was very silty, rather dark, and at first as I went down, I couldn't even see the whale bones. But as I started to look, I realized that they were everywhere, that they were all over the seafloor, and that the invertebrates and marine life were growing amongst them. And this was eerie, but it was also inspiring, because I think a lot of what I was looking at during the year were these very large problems that we're facing and trying to find these solutions for them, but often they would feel bigger than us. But I think there was a time when we would think that whaling would never stop, but it did. And it was an international effort to stop whaling. So now we can dive these sites and know that this time has passed. It was a relic of how far we have come, but also of how far we still have to go. And from this, I felt hope. So I'm going to bring it back to something I learned during my scholarship year. When I was in La Paz in Mexico, I had the chance to go to Magdalena Bay and Baja, where the gray whales approach the boats and seek interaction with the boats and the people aboard them. But it wasn't always like this. In Baja, we'll go back to that one. In Baja, they used to actually hunt the gray whales in those exact same bays where they now approach boaters to interact. And it wasn't even that long ago. Whaling was only banned in Mexico in 1946. But the gray whales here also fought back, and they would crash into fishing boats and sink them. So not only were the gray whales afraid of humans, but humans were afraid of the whales. So long after whaling, they would avoid the whales. And it wasn't until the mid-70s when a fisherman was out on the water and a whale started to approach his boat, and he wasn't able to get away fast enough, that instead the whale just approached and started interacting. And this man's experience snowballed into what we now know as the whale tourism industry in Baja, where the whales approached the boats to interact. So I think it's really important to see these, because it was only about a generation apart between when they were hunting and killing whales within these bays and this form of tourism that you now see. And I think it's really important to know that human culture can adapt, because at the end of the day, it has to. If we want our future to be brighter, it's us who have to change. So what I learned about myself this year is that I wanted to focus on solutions, of turning science into action and shifting human culture to improve the way that we interact with the oceans and their ecosystems. And I want to focus on this in 3 ways: through policy, so that would be fisheries, MPAs, management, which is citizen science, and policy and storytelling, how to inspire future generations. This year allowed me to explore the diversity of approaches and the unique solutions that communities around the world are working on. And I want the takeaway from this to be that the ocean is a space for everyone, for all people and all walks of life and skill sets to connect and to contribute. But this year would not have happened without the incredible ocean family that invested their time, their energy, their expertise into helping me grow and learn throughout this year. I want to thank my coordinators, Anya and Allison, who met with me weekly and supported me and helped me achieve my goals during the year, as well as to the entire Our World Underwater Scholarship Society, who is a team of all volunteers who dedicates enormous amounts of time every single year to making this all possible. My ocean family grew enormously this year. But I also, I lost a very important member of my family, who's my sister, and she's the one that I dedicate this presentation to. She passed away this year, and she is the inspiration for the theme of my presentation, my film, Juliet, Our Hope, Golbahar. She is my big sister, and she is my example of authenticity, of hope, and of kindness. And for me, that's a really important legacy to carry forward in my life's work, the way that I want to move through life, to remember that in this world we live for a lot more than just ourselves. There's a lot of people who contribute to who we are, and in our turn, we also contribute to the lives of others. So it's really hard to be standing here and saying this, but I think often it was hard because for me, in a year of searching for hope, it was hard to feel like I was losing that hope. But it's often in the darkest times that the light shines the brightest and the clearest, and that's what I have to take away from this, because I had an incredible opportunity this year to learn and to grow, and that's something that will continue throughout my lifetime, and that I hope to be able to share with the global community around me. So with that, a huge thank you to all of my sponsors who contributed, and as well as my hosts. Thank you.
One of Rosie's statements that is playing in my head is reading what the earth is telling us. If we walk away with anything from this weekend, it's that phrase, I think. Thank you. What a morning. Oh, well. I can't see anybody. Is Ted in here? Ted's here. Okay. So I'd like to introduce to you Ted Janulis, who is actually, you won't tell by looking at him, he was one of our earlier scholars. And Ted grew up and eventually became a finance guy, but then also became the president of the Explorers Club. Has moved on from that position, does other stuff there, and has actually created an entity that looks at companies that try to solve issues for the planet through ocean work. And he spearheaded that himself, and that is, he can hopefully tell you about the website and all of that, but Ted is representing the Explorers Club today. There's a very close relationship between the Society and the Explorers Club. So, Ted, come on.
So 2 words, Rosie. Wow. That was really something. That was really something. So as Michael said, I was the 1981 scholar, and I've heard that year often referred to as the last year that the selection committee made a truly horrible mistake. Some may have said that when I got elected the 43rd president of the Explorers Club a few years ago, 2 presidents ago. But honestly, I feel fortunate to have been in the middle of both those organizations. And while I happen to have been the first, I am hugely confident that I will not be the last. And I bet that there's someone in this audience now that's going to do that as well. Both organizations have had a huge impact on my life. One example being Paul Schutt, and it's just so wonderful to see that recognition through the generosity of his wife, Linda, that will keep his name in front of us as well as his spirit. Paul was like a second father to me during and after my scholarship year. And among his many contributions, he enabled me to pioneer the concept of the scholar double dip, whereby, like a squirrel, the scholar or intern tucks away undone experiences and relationships. And so when you get to the end of your year, you absolutely have to keep going. So I think I was the first to add on a second year that way. If you want to see some tips as to how to do that, if you're new to the program, you can go to my site, www.keepthepartygoing.com. But those communities, Our World Underwater and the Explorers Club, share so much in common. They share a love of the oceans within their communities, the endless, boundless curiosity about the world around us, and then the storytelling that goes with that. But really importantly, they share a foundational sponsor in Rolex. And I know we thank them a lot, but I just have to tell you, having been involved from both of those sides over a number of years along with Michael, we just wouldn't be here without them. They're just extraordinary in terms of their support. So a few years ago, as you may know, they suggested that the scholarship move its weekend to the beginning of what the Explorers Club had been calling World Oceans Week. And shout out to Steve Barnett for navigating that union, because it's so much work. And it's already yielded catalytic results in that 14 of the people in this audience are going to join 21 other emerging ocean leaders for the next 4 days for our second annual Blue Generation program. And that's people from 12 countries, peer-to-peer learning. They're going to go to Billion Oyster Project. They're going to learn about the blue economy. They're going to learn about sustainable seafood, ecotourism, et cetera. And that only happens because of the spirit of rolling up your sleeves and doing things together. So a huge thank you to them. So with that in mind of bringing us close together, I would also say that there's a thing going on right now where New York City has a climate week in September, and it aligns with the UN General Assembly, and it's a huge, massive thing. Over the last few years, you can see that beginning to happen with the World Oceans Week concept globally. So this year, Milan will have its second. Washington will have, they've been doing it for a number of years. Mexico will have one, Brussels will have one, and ours is getting bigger and bigger. So you're also part of this idea that if we make this a hub and a convening spot, we can make this a more and more powerful thing with real, meaningful, measurable results to it. So with that in mind, I'm so thrilled to be able to work with Mike to give out the thing that the Explorers Club values above everything, which is merch. Swag, sorry, yeah. Swag. We used to do certificates, but they said, hey, the hats and patches are cool. Let's do that. So each of our recipients, and I don't know, do we have a list of the names? Or are they all going to come up at once? They're going to come up. Okay, come up. 2023, yeah.
Will the 2023 Scholars and Interns come
Like he said. 2023. 2023 Scholars and Interns. And you're going to get these very fashionable World Oceans Week hats, and you're also going to get a patch. And welcome to, you get a student membership to the Explorers Club, and we hope you'll all learn more about the Explorers Club. I'll be giving a tour about 1 o'clock today if people want to see Indiana Jones's original whip, or the Apollo 13 flag that never got unwrapped, or President Roosevelt's application for membership, in which he listed being President of the United States 4th. welcome to the Explorers Club.
Thank you, Ted. And Ted, I don't have one of these, so I'm stealing it. Okay. Folks, our incoming scholars and interns. Okay, you can now go sit down with your new hats and go sit down. Go ahead. So this is actually amazing. We're ahead of schedule. So I'm not going to belabor this, but I do want to say one thing here, that we are very grateful to Rolex, but Rolex provides support for the scholarships. The Society has been doing many other things since it was first created. Next year is our 50th. In supporting internships and other projects and such. So if you find it in your hearts and wallets to help support the Society, we could use it. We actually operate at a small deficit, and we have many other projects we would like to build on, and we need support to do that. So the misperception out there is that we are wealthy, happy, and well-funded, which is not true. So spread the word that the Society could use the help, and we would be very grateful for it. So on that note, if Steve or Vin or anybody else thinks I should be saying something else, or you have something else to say. Okay, and just to reiterate, OWUSS, the Our World Underwater Scholarship Society, is an all-volunteer group. So for the last 49 years, the people who have helped to run this, the board, the officers, no one has been paid a dime. So this is a labor of love, and that love could use support. So thank you very much. You're out. Okay.